This program is for the Bachelor of Science third year students. Subject is Botany, semester 6. Course code is BOC 108. Course title Cytogenetics and Plant Breeding. The name of the module is Position Effect. It's from Unit 6, Alteration in Chromosome Number and Structure. I'm Ms. Lynette Fernandez, Associate Professor, PESS, Ravi S. Naik College of Arts and Science, Farmagudi, Ponda, Goa. This module deals with stable position effects and variegated position effects. It explains position effect involving the bar eye locus of Drosophila and understands variegated position effects in the eyes of Drosophila. Any phenotypic alteration as a result of the chromosome rearrangement involving the change only in the order of the genes in the genetic material and involving no change in the amount of genetic material is referred to as position effect. So here there is a change only in the order of the genes without involving any change in the genetic material itself. Position effect may be an outcome of chromosomal aberrations like deficiency, duplications, inversion, and translocations. In deficiency, a part of the chromosome might be deleted or removed or lost. In duplication, a part of the chromosome might be added. In inversion, a part of the chromosome might be inverted after 2 breaks, and in translocation, a part of the chromosome might be removed and translocated elsewhere in the genome. So, in position effect, there is no change in the nature of the gene. So, there is no gene mutation which is involved. Position effect is due to the shift of loci from one position to another or due to the shift of another segment in the vicinity of the locus, due to which the expression of the gene changes. Shifting of loci from one position to another brings about a change in the linkage arrangement on the chromosomes. So, position effect is a change in the expression of a gene due to the changed linkage arrangement. Position effects are either stable or variegated. Stable position effects are uniform phenotypic effects that result from changes of specific segments of chromosomes. Variegated position effects result in a mosaic pattern of a trait usually evidenced in a particular structure or area of the body. Stuyvesant in 1925 analyzed the bar locus of Drosophila. The bar phenotype is a sex-linked dominant gene mutation that reduces the size of the eye. Normal wild flies have round eyes. The X-linked mutation bar caused the eyes to be small and slit-like in males. Males are hemizygous. And in the case of homozygous females also. The female heterozygotes had kidney or bean-shaped eyes, and the flies with the double bar mutation have even smaller eyes. So the normal wild type phenotype showed 779 facets in their eyes. Heterozygous bar eye showed 358 facets, and homozygous bar eye showed about 68 facets. And another phenotype, double bar, or it is also referred to as the ultra bar mutation, they have even smaller eyes and the number of facets are even smaller. The bar mutation was somewhat unstable and it tended to revert to the wild type spontaneously. The bar mutation was somewhat unstable and it tended to revert to wild type spontaneously. Females homozygous for bar produced progeny with round eyes, that is wild type, at a frequency of 1 in 1000. They also produced a more extreme type of ultra bar. They also produced a more extreme type called ultra bar with only about 24 facets, although with a lower frequency. Now, pure ultra bar also produced wild type with the same rate with which ultra bar was obtained from them. They also may give rise to bar. Such unexpected phenotypes in the progeny could be expected due to spontaneous mutations. But it was observed that by Stewart that the frequency of these events was much higher than that would be expected due to spontaneous mutations. So he formulated a hypothesis and confirmed that new types arose due to unequal crossing over in the region of the bar locus. If the homologous chromosomes do not line up exactly during synapsis, a crossover produces an unequal distribution of crossing over. If the homologous chromosomes do not line up exactly during synapsis, a crossover produces an unequal distribution of chromosomal material. This mechanism is unequal crossing over. Therefore, one chromosome will lose DNA sequences, whereas the homologous chromosome will gain the sequences which are lost by the first. Alfred Stewart found that in every double bar fly, crossover had occurred between loci on either side of the bar locus, as is shown in this representation. Crossover had occurred between the loci on either side of the bar locus. Now, this is evident from the recombined marker genes for forked bristles and fused veins. So, this resulted in two types of chromosomes after the crossing over, that is, one with ultra bar and the other with wild type phenotype. He suggested that the change to double bar was due to unequal crossing over rather than due to simple mutation of one allele to another. Later on, an analysis of the bending pattern of salivary gland chromosomes was done by C.B. Bridges in 1936, and he confirmed Stewart-Wendt's findings. He found that the 16A region, which apparently contains a gene for the eye shape, had been tandemly duplicated. And tandem triplications of 16A were also observed, which resulted in double bar. Two 16A regions in the X chromosomes gave normal type of phenotype, that is wild type of phenotype, with about 779 facets. Then three 16A regions produced heterozygous bar eye with 358 facets. 4 16A regions produced homozygous bar eye and 68 facets. And in the case of heterozygous ultra bar also, there were 4 16A regions, but the number of facets that were produced were 45, and the eye was much thinner. It was much thinner or much finer. Okay, a bar zygote and a double bar wild type heterozygote both have 4 copies of 16A region. So it would therefore be reasonable to expect that both genotypes would produce the same phenotype. However, bar homozygote has about 70 facets in each eye, whereas the heterozygote has 45 facets. So this shows that not only the amount of genetic material but also its configuration determines the extent of phenotype. Another example for position effects, that is variegated position effect, is observed in the eyes of Drosophila. Specks of different colors may occur in the eyes of Drosophila due to the rearrangements of the white eye locus. Inversions and translocations can take place in the white eye gene, that is the W+, that is the wild gene for red eye color, and they can translocate this gene to the heterochromatic region, and this may cause white eye variegation or mosaic pattern for the eye color. X-ray treatment caused the X chromosome in some of the flies to break in two places. The middle piece got inverted. The middle piece got inverted and the chromosome ends were rejoined. One of the chromal breaks occurred in the heterochromatin surrounding the centromere, and the other break occurred in the euchromatic region near the white eye gene. The W+ gene makes a product that is necessary for the deposition of eye pigments. When this gene functions normally, it results in normal dark red eye of the fruit flies. But if it is inactive, then the fly's eye is white. Now, as a result of this inversion, this white eye gene, that is W+, was moved from its normal position and it gets shifted to the heterochromatic region, towards the heterochromatic region. Its normal position was in the euchromatic region towards the end of the chromosome. Now, because it has got shifted towards the heterochromatic region, that is the new heterochromatic breakpoint, here the action of the gene may be depressed when transferred to the heterochromatin, or the gene transcription is suppressed, in other words. So, in this position, the gene is expressed in some cells and it is silenced in the others, due to which in those cells where it is expressed, it will give red coloration, and in those cells where it is suppressed, it will give white coloration. Therefore, in this position, it will show a mosaic pattern. The eyes of Drosophila will show a red-white mosaic pattern. From these two examples, it is evident that just the change in the position of the gene loci can bring about the change in the phenotype. This is referred to as the position effect as a result of the change in the position of the gene loci. These are the references, web links. Thank you.